So today we're going to start with the first part or the day one session. This session will be dedicated to the databases only. So we have to take up which kind of databases. To be precise, it will be NCBI, Pubmed, Mesh, PDB, Uniprot, and PDBSUM. So these are the databases which I will be covering today. See, whenever I am exploring database, I will always define a query first. Okay, so let us define the query and then we will do the exploring part in order to find out the information from these databases. So the query which I will define for today's session, let's say that we want to find out.
Sorry, ma'am.
So we will be finding out the research work done on insulin. This is the subject. So I am using insulin as my subject. Okay. So I am trying to find out the research work done on insulin related to treatment for diabetes, any type. This is my first query. Then I want to find out the protein and gene name. Then I want to find out the sequence of protein and functions. Which protein? Insulin. And then I want to find out the best possible structure for human insulin. So this is something which I need to find out. Okay, so these are the queries which I will be trying to figure out using the databases mentioned above. Fine.
Yes.
And open NCBI. NCBI. Okay, so we are opening NCBI. Yeah. So we will be starting to work with NCBI. Okay. Now, before I start with NCBI or any other database, I would like to explain you that whenever we are starting to explore database, we should always understand the pattern in which the database is giving you the information. For example, if you just see NCBI right now, it is divided into 3 different sections. The middle section giving you the result, what you are looking for. The right hand side will be giving you the related searches, something that can lead forward your research or literature mining. And the left hand side is helping you out to filter out the data. You're having vast data, you can filter out on the basis of the left hand side. So right now I wanted to focus on the home page. That is why you can see the middle page is NCBI home. So this is the result. The right hand side tells you that you can also search for Pubmed, Bookshelf, Blast, etc. in NCBI. And the right hand side, and the left hand side is showing you the filter that if you are only specific to certain subject, then you can study the subject from here. There are so many resources available with NCBI. If you just click on all resources for a while, all resources.
Yes.
Yeah. So if you see all resources here, then you can figure out that this all resources is giving you everything which is available here. Okay. So if you just scroll down, you see that all the names of the databases which are present in NCBI are given to you here, ClinVar, etc. Similarly, all the tools which are there in NCBI will be listed down here with a little description. All the kind of submissions you can make it in NCBI again with a little description, they are mentioned here. So this means that if I just go through this all page over here, then in that particular condition, I will be able to figure out that in future, if I am interested in specific thing, I can select that part and use it. For example, I say that I want to study about clinical variations. So I know that if I've studied this particular subject, I can go to ClinVar database to get the information. Okay, so I can do that. Now, in this particular regard, you see that it is clearly given that ClinVar is a particular database that is a source to provide a public tracked record of reported relationship between human variations and observed health status. So I know this thing. So in future, I will be going to ClinVar database for the clinically reported variations in humans. So I know this thing. How? Because I have gone through this. So first suggestion is that go through this particular page and read all the abstracts or the gist given to you after each of the databases, tools or submissions, etc. Okay. So every database we will be using, they will have their own patterns to make you understand how they are giving you the data or how they are managing the information. Okay. Scroll up. Now, if you scroll up, now this all page is having everything. First, they will mention database, then tools, then standalone versions of the tool, etc. If you talk about you want to only study about the tools, then you can simply go to the tool section over there. If you want to go to databases, they will give you only the list of the databases. Okay. And if you want to find out only those databases, supposedly my focus for your session will be proteins. So if I only want to study about the proteins, then you can put a filter from the left hand side, which is proteins. So then everything which will be there on the screen will be associated with proteins. Please click on protein. Okay. Now if you see all databases, it is the list is less. Why? Because now it is only dedicated to proteins. Some or the other kind of information associated with the proteins are available in these databases. That is why they are over here. Okay. So right now I'm not focusing on any one, although I want or I am interested in protein information, but still I am not going into that. I want to show you a general way of finding out the information when I have no specification. Okay, but you can specify, you can narrow down your query from here. Fine. Coming back to the home page, NCBI home. So I will be starting with my query and I have nothing in hand. I don't know that which name or specific keyword I should use. So I'm going very random, which is my protein name, and that is insulin. So I will type insulin and say click search. So you see that what this NCBI does is whatever information is given in NCBI where insulin is mentioned once, whether it is insulin growth like factor, whether insulin receptor is there or anything for any organism, it is over here. They have bifurcated the information into different sections like literature, genes, chemicals. PubChem is basically the chemical, genetics, genomes, etc. So you can pick up from here also. Like I said that I don't want to go with the specification which was given previously in the all sources. I prefer to go over here because now today's focus is on literature. I want to collect the literature first, right? So which section I will be dedicating today is the first section, the literature part. Now literature can be books, literature can be the mesh is medical subject heading, which I will be doing it with you. It can be the National Library of Medicine catalog, it can be Pubmed and Pubmed Central, PMC and PME. Now these particular sections can be used to get the data which in the starting I wrote as my query. So it is very important for you to define what you need to find out first and then start with this. So I know that I want to do the literature. So my particular section is literature today right now. I want to study about the proteins and I will be going to the protein section and see what I want to study. I want to study about a particular conserved domains in the insulin related stuff, or I want to go with the structure or anything like that. Okay. So you can do that. So let's go back to the literature, and today I am focusing on PubMed and mesh. So mesh I will be taking a bit later. Now you see that if I focus on PubMed right now, the same way you can explore the other things as well. Now if you focus on the PubMed, you see that there are 4,16,380 articles available. Now it is very much difficult for me to read out all these articles to get the information, right? It is not impossible, but it is very much something which is not appreciated, that as a bioinformatician I'll be just reading out way. So what I'll do is I'll just right click open PubMed articles in new tab.
Okay.
Okay, let it be. It's okay, no issues. Okay. So we are having 4,15,300 and something papers. Okay. Now let me just make you. Now you need to understand PubMed right now. The PubMed is divided into two sections here. First section, the result, and the left hand side is basically talking about the filters. The first colorful, the green graph, which you can see, that is helping you out in order to understand that how many papers were published year wise. If you just move your cursor on it, just yeah, you see that in 2009 14,000 papers were published. So this way you can also do that. You know that whenever we are collecting the information, we prefer that we pick up the recent articles. So 5 to 10 years articles I can do. So you can just drag that 1922 wala circle to 2015 or 2012, something like that. Okay. So how many articles we are left with?
1,03,000.
Right. Okay. Now we can also put filters. Now if we scroll down, we see that text availability option is there. Text availability means you're having abstract, free, full text, etc. Now this is a particular thing. Yes, you can do it. Filtering of your data with this, but I prefer not to do this. Why? Because then my research will be biased for data available and the non-availability of the data. But as a researcher, if I feel that this paper is very much informative for me, but it is not available in the public domain, does not mean that I will not be reading out that paper. Okay, there are so many options to get those articles. First, which is very much legal, which is what? You can ask the authors to give you the copy of the manuscript first, or the draft of the paper. Second, you can go to Sci-Hub. Okay, now Sci-Hub is something which is not legal. As a researcher myself who is publishing work, I also care about that. But sometimes we are not, it is not possible for us to pay for every paper. Okay, so in that particular condition, I use SCIHUB in order to find out the papers for which the free articles are not available. That means the entire paper is not available. You can try for SCIHUB. Now it is 5% chances. This is a very maximum chance I'm keeping up. 5% you might not get the paper over there also, 5%, but 95% chances you will get all the papers over there. Okay, we cannot cite Sci-Hub anywhere. We always have to give the reference to the articles only. No Sci-Hub should be mentioned in the research. So text availability is something which I don't want to use as a filter because that will restrict my research. Associated data, something not of my type right now. I want to be focused on insulin, so I don't want associated data. Article type means you want to study about the review papers only, or you want to study about those papers which are mentioning some or the other information or research related to clinical trials, meta-analysis, or something like that. I am not interested in that also, because see, when I'm collecting the data, definitely review articles are the best way to start with. Why? Because review articles summarize a lot many research work. Okay, so in that particular regard, yes, review papers are helpful, but initially I cannot put those kind of filters because I might not be having a review paper which is compiling XYZ paper, so I might skip good papers in that case. Coming to the next filter, which is publication date, which is 1, 5, and 10. I've already put the filter to 5 years last in the starting. Okay, so you can click over here also 5 if you want, or 10 if you want. Okay, there are additional filters which you can see right now. Now additional filter will be associated with species, article types, etc. Now these are the other things. Now in the article type you can click on, like, the papers on the basis of the journals, the papers on the basis of the clinical study. You want to only study trials, phase trials, and etc. You can put the filters. Okay, now this is very specific based on your requirement. I don't have any requirement for now, so I will not be able to put any filters associated with this. Let's go by species. I can put a species because right now I'm looking for humans. I can click over here. Yes. Then I have nothing to do with the language right now, sex, subject, journal, age, nothing. I'm not interested. There are certain conditions where the research are age dependent. Then you can put age, sex in that particular places as well. Okay. I don't have any specification for now, so I will click on show for now. Okay. Now in this particular, scroll down, scroll down. Yeah, now click on humans. So we have added that particular additional thing over here. Now we are putting humans. So how many papers we are having from 4 lakhs? We have shortlisted the papers to 40,636. Although I have not put much filters to this, but definitely I may put those filters if I'm very specific about any article type or information or data which I want. So now I am left with 40,000 papers. So what I'm supposed to do is I'm supposed to read out the articles. Okay. I'm supposed to read out the articles here. Okay. So it becomes again 40,000 is comparatively not a big number as compared to 4 lakhs, which I was having in the starting. But this is not something which is feasible again as a bioinformatician. So what I'll do is the longest way is read out the articles. First, the abstract. If you find it relevant to your work, you read the whole paper. If not, then proceed further. So that is a big task. So what I will do now is I will use a particular refinement. I call it as a tool, but it is a database actually, which is MESH, Medical Subject Heading. Okay. So I will not use it from here. We'll open a new tab and MESH, MESH, Medical Subject Heading. Now this is a database with NCBI. Now the use of mesh, single M. The use of mesh is what? It helps you to refine your literature search or Pubmed search to be precise. So whatever papers you have got there, you can also put a very second level of filter using the mesh. It helps you to work on that. So let us type the same thing over here, insulin. Enter. Now what you can see on your screen, these are not your articles or paper, these are categories in which the papers have been put. And associated data is also given to you with each category. Like the first category says that if you want to read about those articles only where you want to study about 51 amino acid pancreatic hormone that plays a major role in the regulation of glucose metabolism and associated with some diabetes and all. Okay, so if you read this particular gist, you see that this is what I am interested in as well. So this means that this is the entry of my interest, but every time first entry is not your related entry. You have to read the abstract or the gist given to you. If that suits you, then you will be proceeding further with the entry. If not, go ahead with the rest of the entries. Find out the one which is associated or relevant to your work. For my case right now, the first entry is the one which I will be going through. So click on the first entry. Once you click on this first entry, you will be getting a lot of subheadings over here, which you can see like administration, dosages, etc. Now you see that how specifically this tool will help you to find out only the treatment. You remember, I want to find out literature related to insulin treatment and other things, right? That was my research. So you see that you find out some subheading associated with your query. So let's say therapy is there, therapeutic use is there. Okay, so you can select multiple headings. You can select all of them also. But I am using this mesh to narrow down my search. Okay, sometimes you have more than one requirement. I want to find out the therapy, I want to find out, or I want to study about the genetics also. And I want to study about the analysis or anything. I can select many, but as per my experience, it's always better to bifurcate the papers in categories. First, gather the information about therapy, then gather the information about genetics. So that becomes more systematic. So you can select multiple, but I would suggest you that always pick up less subheadings because we are trying to narrow down. If you put so many filters over here also, so not filters, so many subheadings over here also, you will get again a big data. Again, you have to, pick up which paper is giving you that. So I'm just going by the therapeutic use and therapy. And then there are two check boxes mentioning restrict to mesh media topics and do not include. So I will click on the first one for sure. Now, what is the second one? Scroll down. Whenever the data is submitted in mesh, they categorize into hierarchy. Okay, scroll down. The hierarchy is given to you here. Yes. The black bold insulin which is written, that is where your query lies, and above and below. So if you click on that second checkbox, it means that they will not give you any paper in which belong to the categories below the insulin. That means insulin, regular, human, isophane, insulin, regular, pork. These are the terminologies which will not be considered in order to filter the PubMed search. Now, this is very, very specific. Now, right now, I am not sure that whether this regular, insulin, human, isophane, etc. is relevant or not. So, if I'm not sure, I will not click on the checkbox which is below that. But if I'm very much sure that I'm not interested in these titles, I'm interested in everything above the hierarchy, then in that condition, I may check the second checkbox. Okay. So you have to be specific. If not an issue. We will be reading out the data and proceeding further. So I am not clicking on the second checkbox since I am saying that I am not sure about the topics mentioned below the hierarchy. So I will go up. I'm just clicking, but clicking or checking the first checkbox is must, so that whatever data you get in this category will be filtered according to the subheadings which you have selected. Then we go to, on the right hand side, we have add to search builder. Right hand side. Click once on that. And then search PubMed. So how many papers you're getting? 19,000 papers I'm getting, right? Now in this also you can put filters like we have put the filters in the simple PubMed. Okay, so I can do that. Scroll down. We can make it 5 years. We can make it human. Scroll down. So how many papers we are having now? 2,534. So you see that compare the first tab and this tab, from 40,000 specifically, I am having only 2,534 papers. So this way, the mesh helps you to get or narrow down your queries to specific subjects. Now you will be reading out the paper. Now there is no option available over here. Okay, you can start then with the filter that you start studying with the review and other things. You can put filters to that, but this is the common filters which you can apply to all. Okay, now you see that treating diabetes, let us say that first, read the title. It says treating diabetes 2, diabetes type 2 in COVID-19 patients. So this paper is associated with COVID-19. So I am interested in COVID-19? No. So I will not be talking about this right now. Then go for the next title. So title is basically what? The abstract of the paper. Okay. I would be more precise. I would say it is the abstract of the abstract. So title, if you find it relevant, go with it. If it is not, proceed with the next title. So title should be relevant to your work because titles are written in that way. Okay. If you are interested in the first paper, go with it. Read out the article abstract. If it is relevant, proceed with the full paper, else not. Okay. So obviously, I'll not be reading on the papers over here. Now, if you read, supposingly you are starting to read with first paper, okay, any papers over here. So let's say first, okay. I am not interested in COVID right now, but still, I'm assuming that this is a paper of my relevance. Now here, when I read this particular paper, I'll keep on reading it and I'll find out when I read this article, I will get to know what is the exact name of the protein insulin. Because I started as a random, I know, okay, this is insulin. Let's say I read the paper and they say that XYZ is the name of the protein. So I will write down that name over there. So protein may, I will be writing down Insulin is the name of the protein. For that, you have to read the articles, but obviously. Okay. So when you read the articles, you'll get to know about the name of the protein. You might get name of the gene also, which is INS. So this data I'm talking about when I have read the articles on insulin, diabetes. Okay. So I know this data from the papers. That's INS and insulin. Okay. After getting to know about the proteins and the gene names, I can proceed further with specific databases like PDB, Uniprot, and PDBSUM. Okay.